All right, buckle up, peptide pals. Kai Rivera here, your chief investigative scribe. If you're passionate about cutting-edge research, you absolutely need this vital information immediately. We're diving deep into something that could revolutionize our fight against drug-resistant bacteria. For ages, we thought our body's own antimicrobial peptides, or AMPs, formed static holes in bacterial cell walls. But new research is suggesting a much more dynamic, fleeting interaction. This isn't just a minor update. It's a complete paradigm shift for understanding these powerful natural defenses. Ready to uncover these game-changing insights? Let's dig in. First, let's address the critical issue: antibiotic resistance. It's a growing global research challenge. Bacteria are evolving, shrugging off traditional antibiotics. This presents significant research hurdles. Millions of resistant infections occur annually, highlighting an urgent need for new research. The CDC reported over 2.8 million antibiotic-resistant infections in just 1 year. The WHO designates antibiotic resistance as a top global public health research threat. Finding novel approaches for infection control is not just important, it's critical for research. This is where antimicrobial peptides, or AMPs, enter the research spotlight. These tiny protein fragments are nature's broad-spectrum defenses, found widely in research subjects. They represent a natural immune response, a first line of defense in many organisms. For decades, the leading theory was AMPs created static pores in bacterial membranes. This puncture-and-kill method suggested a simple, straightforward bacterial elimination. The Barrel-Stave model envisioned AMPs forming stable, tunnel-like structures through membranes. Alternatively, the toroidal pore model described membrane lipids bending to form pores. Both models focused on permanent holes, leading to cellular contents leaking out. Researchers utilized techniques like NMR to observe these supposed stable structures in action. However, some experimental pieces didn't fully align, creating a somewhat static research picture. It was like trying to understand a dynamic process from only a still, incomplete image. And that blurry, static picture is precisely where the true plot twist begins. We thought we understood the fundamental mechanics, but science, ever so wonderfully, keeps evolving. Before we reveal this groundbreaking new understanding, a quick shout-out. For more deep dives into fascinating peptide research, make sure to follow me, Kai Rivera, on all platforms, and subscribe to peptides.today. We're always unearthing new gems. Now, prepare to have your assumptions about AMPs completely overturned. A recent study published in PNAS dropped a major bombshell on our understanding. It suggests AMPs cause dynamic, transient water channels, not static, permanent pores. This means the bacterial membrane isn't just punched, it's constantly disrupted and shimmers. Imagine a crowd at a concert. AMPs create momentary gaps, like tiny mosh pits. These fleeting openings allow essential ions and water to leak out rapidly. The bacterial cell cannot maintain its internal balance and thus ceases to function. No massive stable hole is required. It's a death by 1,000 dynamic disruptions. This dynamic mechanism clarifies why AMPs work at low concentrations effectively. It also explains why bacteria don't quickly develop resistance to these peptides. If the target is dynamic instability, bacteria struggle to learn how to fight it. Broad-spectrum activity also makes sense. AMPs target general membrane disruption, not specific receptors. So why does this groundbreaking understanding matter for future research and development? Knowing this mechanism allows us to design smarter, more efficient AMPs for study. We can fine-tune chemistry to maximize the mosh pit effect, enhancing potency. This approach could help overcome resistance, as membrane rebuilding is a tall order. Engineering targeted AMPs is also possible, disrupting only bacterial, not human cell membranes. The future of infection research is fluid, dynamic, and powered by innovative peptide insights. This truly changes the landscape for designing a new generation of antimicrobial treatments, giving researchers a crucial advantage in this ongoing discovery war. It's a prime example of nature always having more fascinating tricks up its sleeve. Before we sign off, make sure to follow me, Kai Rivera, and Peptides.today for more unearthed epic research. And remember, before we sign off, a quick legal disclaimer. This podcast is for informational purposes only and is not medical advice. All compounds discussed are for research use only and not for human consumption. Always consult a licensed healthcare provider for medical needs and treatment.